Good morning, good afternoon. Happy to see everyone. We are here in Tunis in the middle of Ramadan, exactly the middle of Ramadan, and this is the 18th lecture of the Logical Reasoning in Human Genetics course. Today we have the pleasure of welcoming Dr. Jesus Melgarejo, who is Assistant Professor of Neurosciences at the University of Texas, Rio Grande Valley. He will be talking about variability in biological systems. As you know, this lecture or this course, the Logical Reasoning in Human Genetics, is a partnership between Columbia Global Centers Tunis and Institut Pasteur Tunis. So we also have several members of the steering committee for this program. And now I leave the floor to Dr. Terwilliger to introduce Professor Melgarejo even more, and also and so that we can start. Thank you.
So Jesus has been a colleague of ours for many years. As we talked about briefly a few lectures ago, we started doing a study in Maracaibo, Venezuela, of two different populations in the city about 25, 30 years ago now, I guess, or about 30 years ago. And the initial purpose of the study was to look at genetic factors related to aging and memory decline. And so along the years, we did a lot of repeated sampling of a number of interesting measures. And Jesus was one of the bright young guys working in the lab in Venezuela. And he came to spend some time with Dr. Jolie for a while at Columbia University, working with us on different aspects of what to do with this project. Now, while the project itself, we have genetic data, and the main purpose was eventually to look at the genetics of aging-related traits, what Jesus looked at was something more about just analyzing the traits themselves, and we still haven't gotten to the stage yet in his analysis where we have included the genetic factors in the analysis, but these are all things that will happen in the future. And so we thought it would be really useful for you to hear, well, first of all, to see what a successful scientist this sort of a project can produce. If you set up your own project there in Tunisia that are really well designed and interesting, you'll have many people coming through that can use this to build a great career. And Jesus is one person who's done that. So I'll let Jolie introduce him personally, but I just wanted to set the framework for what got us to invite Jesus here. Even though this talk is not specifically about genetics, it's coming out of a project that was originally about genetics and will be in the future. So, Joe?
Okay. It's a great pleasure to introduce Jesus. I have known Jesus for about a decade. When he was a young medical student, didn't know anything, but incredibly enthusiastic about learning and doing research. And I have known him for so long. And he spent a summer at Columbia to learn various genetic epidemiology methods. So, and that was the beginning of our collaboration. And since then, he went on to, after he got his medical degree, he got his doctorate degree in PhD in, I guess, biomedical sciences at Catholic University of Leuven in Belgium. So he's legitimate card-carrying physician scientist. He's been collaborating for a long time. And when he's not doing science, he plays guitar. So there's some music trend here in both Joe Terwilliger being a tuba player and him being a guitar player. So unfortunately, I don't play anything of any sort. So that's about all I can say. And Jesus has done a nice job of, rather than looking at a disease or one methods, he's looking at physiological system to see how the physiological system impacts various diseases in different organs. So he looked at eye diseases, and then he extended that to Alzheimer's disease. So I think he's one of the pioneers looking at how eye disease are related to neurological dementia. So I think he's been quite innovative, and he's done some nice work. and hopefully we'll continue to collaborate. And without further ado, here is Jesus. Go ahead.
All right. Well, Professor Jolie and Dr. Williger, thank you very much for your introduction. And yes, it has been about 1 decade since I started working with you guys, and you have been very important in my guidance to up to the point that we're going to discuss today. So I really have to thank you, and I hope that that's true. That I hope that we can collaborate in the future, because there's a lot of things that I believe there still need to be done, and I hope that I can keep working with you on these topics. So I will also like to thank the organizers for these events. I feel really happy and privileged to be here giving you this talk today, and which is about variability in biological systems, a study of the 24-hour blood pressure monitoring. And before I go to the whole picture of what we have been doing, I had a slide which I wanted to show you in my experience, which I believe what Professor J.T. Williger and Jolie described, at least for my side. My introduction in the world of science had followed different steps that we know that there are some steps that we need to follow, that there's just procedure that we need to do in order to get what we would like to do. But I wanted to share my experience. So we know that our first involvement in research has to do that we find a lab, we find a person that is doing this research, and once we get involved, we're going to pick a project, or, well, we get assigned a project, a topic, and that we started working on, and we started developing the topic. And eventually that will lead to a product that could be a manuscript, that could be an abstract, that could be a report. But initially we get involved in one topic. Once we finish with that topic, we also, at least in my case, then I got an opportunity to be introduced to a new topic. And once I finished that topic, which at that moment I thought, hey, this is not related with my previous topic, I got a new one. And then when I got there, I was like, really? Is this what am I going to be doing? Does this have any relationship to what I had before? But actually it did. So, and I will tell you that later on. And even after I keep working on that project and I found that there is some link, then I asked myself, what am I doing? Why am I doing this in this way? And now I wanted to look behind and see other questions related, just to explore. Because eventually with that exploring was what led me to one of the topics that I'm going to be discussing to you today. But if I stayed only in the one question that I was assigned to, I think that would have impacts my research project today. So this exploring part was really important to me, even though I didn't know how to do it. I just wanted to explore. Then the collaboration, as well, if you have noticed, Dr. Jolie and Dr. Williger, they have been mentor to me, and also who is not here today, Dr. Gladys Pasteur. She's my supervisor here at UTRGV, and she has been my supervisor since I started the neuroscience laboratory. And not only them, but also people outside our group, where I did my PhD, where I went to do my fellowship, that's really important. That whatever that we are doing, that we are sure that we are collaborating with some other people, because the kind of work we do, we cannot do it alone. Probably there are some research that can do it alone, but I know at least in our field that we need to have a multidisciplinary team, not only to develop a scientific idea, but also because it will guide you. And as I was a young researcher, with no experience, I needed to learn a lot of things. I still need to learn a lot of things. This part was essential. And of course, in the way that you are learning from them, they will also give you the opportunities they will introduce you to these topics, and they will give you the chance to start exploring. So this part was also essential. And the experience part, in which I know there's still a lot to do, but even when I tried some hypotheses that didn't make any sense, all of that, that gave me experience. Even if it didn't work out, what if my hypothesis, what I tested was something different to what I thought before, that gave me an experience that later on applied on my projects. So this is kind of how, in the summary, how my process has been, and I will tell you later on how does these specific steps fit within my research development more clearly. So now let's start with the talk. When we talk about variability in biological systems, and specifically 24-hour blood pressure monitoring, why we started looking into this? Well, because we were interested in a disease that is called glaucoma disease. The disease is the first cause of irreversible blindness in the world, and is characterized by a retinal ganglion cell bed, that is the cells in your eye that they capture the visual stimulus and send it back to the brain. So these cells in the eye, they start dying, and what we're going to have is a clinical presentation of vision loss. And this is progressive, chronic, and irreversible. Nowadays, the only clinical treatment to treat the disease is to reduce the intraocular pressure. That is, this picture here, we have the eye. The yellow layer represents the axons that communicates the cells, the neurons to the brain. And inside the eye chamber, there's liquid. This liquid, there is a mechanical hypothesis that when the liquid increases, it exerts a mechanical compression to the optic nerve, and that eventually causes retinal ganglion cell death, and with that, the progression of the disease. But researchers and ophthalmologists, eye doctors, they noticed that reported that about 50% of the cases in which the IOP is normal, it reached a therapeutic range, the patients or subjects, the progression of the disease still progresses. So they thought, okay, we're controlling the IOP, so something outside the eye must be happening, probably. Or we need to look into other hypotheses. And that's what they thought about the vascular hypothesis or pathogenesis of glaucoma. This pathogenesis, this hypothesis states that there is a ischemia in the optic nerve, that is this yellow layer, which is interruption of blood supply to the optic nerve head, that eventually leads to retinal ganglion cell death because the cells are not receiving blood, leading to irreversible vision loss. And the main factor that they found related to ischemia of the optic nerve was reduced or impaired ocular blood flow dysfunction. And one of the determinants of this is what we call the ocular perfusion pressure. The ocular perfusion pressure determines how much, in these words, how much blood does the optic nerve receive. If the ocular perfusion is low, it means that there is a reduction in the blood flow and supply to the eye. So they also identify what are the determinants of this ocular perfusion pressure, and they reported two: the IOP and the mean arterial pressure, or the blood pressure when you go to the doctor. So whatever causes reduction in the ocular perfusion pressure, that will eventually lead to all the cascade that you are seeing here, eventually ended up in irreversible vision loss. When they look into this mean arterial pressure, that is the blood pressure when you go to the doctor, they take your blood pressure, and researchers identify that low blood pressure or hypotension is a risk factor for glaucoma because of this. Why? Because the OPP the ocular perfusion pressure is just, we can calculate it as the mathematical, just the difference between MAP minus the IOP. And here there are two cases in which the blood pressure is normal. We do a simple math, and then we're going to have 75 in the OBP. But if your blood pressure is really low, for example, here 70, then of course the ocular perfusion pressure will be lower, and this will lead to glaucoma progression. But there was another problem, and that's where we came in. That there is blood pressure when you go to the doctor. They take about 1-2 measurements, but that is not the whole picture. Because if I take your blood pressure now and then I take him, in the afternoon or 5 hours later, it's going to be different because it has a normal variability. So we started looking into 24-hour. Well, researchers already did about 15 years ago into 24-hour blood pressure monitoring, and they say, probably because during the nighttime the blood pressure decreases, that might relate to glaucoma. So what I wanted to tell you from this is that looking into 24-hour blood pressure variability, they were interested in variability. And what is this method? It does the same as when you go to the doctor. There is a cuff that takes your blood pressure in your arm, but the difference is that the device is an automated device, that you have a little box here that just put the patient will just go, or subject will go to their home. They will do their normal daily activities. They will not stop doing anything, but the cuff will be taking the blood pressure every 15 minutes during the day and 30 minutes during the nighttime. So compared to the 1 or 2 measurements that you can get when you go to the doctor, now you're going to have about 80 measurements, and this is a blood pressure, and this is a graphic of how it looks. So of course you're going to have a variability in the blood pressure. And if you can see here, during the nighttime when the blood pressure goes down, it's normal because we're going to sleep, and we're in a resting state, and of course our vital signs decreases. What they report is that some patients with glaucoma, this nocturnal blood pressure decreases too much. And again, if the blood pressure decreases too much, it will lead to lower ocular perfusion pressure, and this will lead to all these cascades resulting in glaucoma. But even like that, we started looking, no, it should be more because not everyone experienced an extreme dip in the nocturnal blood pressure. And by looking into 24-hour blood pressure monitoring up to now, in relation to glaucoma, this is the research products that we have been able to publish and generated. So we have about 5 papers that I have authors since, I think we started working on this about 2015, I think, if I remember correctly. And this has been what we have been able to publish. And I will go through each of these briefly, and I will tell you what is the part that we believe is the most important one related to variability. So before I start describing all these projects, I wanted to say in which sample we apply or we tested our hypothesis. So what we did in summary, we conducted baseline or follow-up studies using population or participants from the general population. Just to give you a brief example of what it is, so what we did with the Maracaibo data, for example, was that we enrolled participants from the catchment area. And of course, we're going to have some subjects that they are healthy. Some of them, they are at high risk because they have risk factors. And some of them, they will have the disease. In our case, for glaucoma, we have those in this case, the disease will be glaucoma. But we also did a case control study in which eventually what you do is to match healthy and disease or cases. This for us was glaucoma, and we matched them by age, sex, and other clinical parameters. So in summary, this is what we used to test our hypothesis. And I wanted to show you about the Maracaibo study because this is where all started about this project. So the study is located in Maracaibo City, in this region, in the state of Zulia. And this is where I was born and raised. And the study has been active since 1997, if I remember correctly. And in 2010, there was a new cohort in which we measure, we did iron measurements and also 24-hour blood pressure monitoring. That's, I think it's Professor, I don't know if Professor Willig or Jolie have mentioned before, but in that specific cohort in which we did the eye examinations and 24-hour blood pressure monitoring, which is, this is where they live, kind of where they live, because there was a pedigree that was constructed in which they were able to identify that at least 8 generations there's a 8 generations family in this catchment area, which they didn't know they were family and etc. But there was a family status in which that population was where we did all these eye examinations and 24-hour blood pressure monitoring. And this is the center of our city, and this is also part of our city. Okay, I'm not gonna go with statistics within each project. It's just, I will give you just a summary of what we did. For descriptive, this is what we use. We reported the, for example, baseline characteristics with standard deviation or median or interquartile range if it didn't follow a normal distribution. These were the logistic regression models. We use logistic and linear regression modeling for the cross-sectional studies, and for the longitudinal one, we use mixed models. And we also try to test risk improvement, that is, whatever we identify as new, will that improve the clinical relevance of previous risk factors in order to predict glaucoma? And for that, we use these methods. So in part number 1, I'm going to go really briefly. We identified that if you remember the picture 24-hour blood pressure monitoring, essentially we take the blood pressure for 24 hours. What we were able to report is that those, in the case of there's a high variability in the blood pressure over 24 hours, that relates to the probability of having glaucoma. And in these forest plots, what I'm showing you, it's just 2 types of different analysis based on the visual field effects. But what is of interest is that in both cases, having high variability in the blood pressure during 24 hours increases the probability of having glaucoma. And this is where everything started getting really intriguing, because this is the graphical representation of what we found. Red lines indicates patients with glaucoma. So you're going to see, compared to the blue line, which is our normal eyes, you will see that their blood pressure changes more during the day, during the over 24 hours. That's what we reported. But then, if you remember from the first slides, there is a hypothesis in which what matters to the eye is when the blood pressure goes down. So when we saw this graphic, we started asking ourselves, are we properly interpreting blood pressure variability or variability? Because the graphic, this picture I showed you in the beginning, says that what matters for the eye is when the ocular perfusion pressure goes down. And the only way that this can happen, if we are looking into the blood pressure, is that the blood pressure goes down. So essentially that explains that in this, during these episodes, over 24 hours, there should be the moment in which the ocular perfusion pressure goes down sporadically. It's not constant during 24 hours, but it's just a moment. It's an episodic drop in the blood pressure. But the problem with that is whatever index we use for variability, standard deviation, variability independent of the mean, coefficient of variation, whatever index you use to quantify variability, it does not give you the information, at least for us, for glaucoma, we need, because this is just a statistic measures of the values around the mean. That's all we know. So we have a mean, and then we have the standard deviation or whatever index, and we just know with an absolute value how many data points going to the left and to the right side of the distribution. That's all we know. But for biological systems, this might represent something different, because for glaucoma, what we are interested in is in what is happening here because of those exact moments of the blood pressure variability is when we are hypothesizing that the ocular perfusion pressure goes down. But again, by looking into variability by itself, we are not able to really determine what is happening there. And this is where I started looking into, okay, probably in biology, we have to look differently. If we have too low blood pressure levels or too high blood pressure levels, they might be a cause of the disease or a consequence of the disease. So we need to look them separately. And these papers here, they're published in the top journals in cardiovascular science. We have JACC, and then we have the European Society of Cardiology. And what they are showing there is that visit-to-visit blood cholesterol variability relates to cardiovascular events. What I meant is this. You have visit number one, you go to the doctor, they take your blood, they measure your cholesterol levels, and they measure your cholesterol level for about 5 years, as an example. And what they reported here is that the variability in each of these visits, the higher the visit-to-visit variability relates to higher cardiovascular events. And these results were published in important journals that were somehow applying to clinical practice. And I believe, if you are familiar with, we know that if we have high cholesterol, that's bad for our heart arteries. So I am guessing, because it's not reported or described there, that if I tell you that high variability in cholesterol relates to coronary artery disease, I'm sure that nobody will guess or will say that, yes, if you have high visit-to-visit variability, you will have, the lower your cholesterol levels, the worse your cardiovascular outcomes. No, what it would mean is that if you have high visit-to-visit variability, it means that your cholesterol during each visit is going up, and it's going above the normal level, and that eventually is increasing the risk for you to have coronary artery disease or a stroke, as they have here. But this kind of reasoning is a clinical reasoning, and for that they treat cholesterol levels. They didn't think the other way around of the distribution of the variability. That is when it goes low, the cholesterol level, because that will be good, essentially. But with this reasoning, we have no information about this. So this is where for glaucoma we thought, hey, no, we need to look into variability separately. I don't know how it works, variability. I know that variability is extremely important and discussed topic in genetics. I don't know how this will be applied to that, but at least I know that here we needed to disentangle any index of variability we used in order to better understand glaucoma. So what we did was to, and this is an example, during 24-hour blood pressure recording, we identified the 5 largest peaks in blood pressure. These are the blue dots here. So you will see that there is a drastic increase in blood pressure, and also identify the 5 largest drops in blood pressure. Those are the red dots here. So you can see that then suddenly blood pressure went down, and the same is here. And we separately quantify these indexes, and then we wanted to see their association with glaucoma. So eventually, using the Melgarejo data, that was our first step. We were able to publish that participants with glaucoma, as we have here, compared to healthy participants, when the blood pressure went down, they dipped longer. So drastic drops in blood pressure were longer in those with glaucoma compared to healthy participants, but also the magnitude of the blood pressure drop was also lower in glaucoma compared to healthy participants. And when we did regression models, such by age, sex, and other confounders, we found that only the dips in the blood pressure, that is, these red dots here, only those were related to glaucoma progression, and blips or increases in blood pressure, they were not. And of course, if we correlate this with variability, they are strongly related because it depends on the variability. But if we quantify them separately, this is what we observed. And this is what we're expecting, because for glaucoma, what matters is when the blood pressure goes down. After this, we wanted to move and test our hypothesis using a different cohort and also using, more importantly, longitudinal data. And for this, we built what we call GON, which is glaucomatous ocular neuropathy, 24-hour blood pressure monitoring database. And I am showing you this graphic here, and I think if I remember correctly, Professor Williger, he mentioned that what we discuss here, what you have been discussing during this course, is that you're also able to do research wherever you're from, and that you're able also to collaborate with other people, but importantly that we were able to do research. And this graphic here is just to show you that the data that we were able to we didn't collect, because this was a retrospective study that has longitudinal data. So these were glaucoma patients from the glaucoma department of the UC Leuven Hospital, where I did my PhD. So eventually, glaucoma patients, they are everywhere. They have to go to the doctor. And in our institution, or back then, those patients with glaucoma, in which they suspected glaucoma progression despite controlling all risk factors, they did a 24-hour blood pressure monitoring. So once we were able to identify this, I manually identified those patients, and I manually extracted the data, 476 patients. And I have to tell you that every patient took me around 20 minutes, because getting into the system is slow. We have to look into different parameters, but I was able to do that. It took me around 1 year to build up all this database of data that already existed. That's what I wanted to tell you with this graphic, that probably there are other centers in which it doesn't need to be glaucoma. It could be any other disease in which if we follow the hypothesis and the protocols pertinent, we have to do the ethical protocols to access the data, etc. There's data that we can use in the future to test our hypothesis or to start collaborations. And in the end, this was the product in which we tested our hypothesis using longitudinal data. And this is just a follow-up chart, flow chart graphic of the exclusion and inclusion criteria and the total number of participants. In the end, we analyzed, although I manually extracted the data of all these patients. Then we figured out some non-informed consent, or they had multiple chronic deterioration comorbidities such as cancer. So these are kind of inclusion or exclusion criteria that you might apply. But anyway, this is the data that was already there, about 15 years of retrospective evaluations of glaucoma patients, and we were able to use this data. So eventually we tested the same hypothesis, just using progression of glaucoma longitudinal data of patients with primary open-angle glaucoma. So we have 110 patients, and we have had over 3,000 observations. That means that because these are patients and they are followed at the clinic because of their disease, we were able to have, I think it's at least 10 evaluations or 8 evaluations. I don't remember. I think it's 8 evaluations, a median of 8 evaluations per patient. And this is the number of observations we were able to have. And this is why, if you remember before, I applied mixed model. Anyway, what we were able to show was the same as before. First, that the variability in the blood pressure, that is the variability over 24 hours, the greater the variability, the worse the progression of glaucoma in adjuster and adjuster models. And when we identify separately flips and drops in blood pressure, we saw that only drops in blood pressure, they were related to glaucoma. And these are different indexes here that you can see that they are all significant. So after we were able to replicate our hypothesis in a different cohort with using longitudinal data, then we wanted to test, there are certain clinical parameters that we use in clinical practice to determine risk risk scores, and or that we use for treatment of patients. So we wanted to show that on top of those clinical factors, if we include other indexes such as variability or drops in blood pressure, we might be able to improve the risk stratification of glaucoma disease. So what we did was to build a model in which we have the clinical variables. And when we included the blood pressure level, it didn't improve the risk stratification. But when we include variability and those indexes quantifying the drops in blood pressure, the model improved, the performance in the model improved, and it was about 4 to 7% risk stratification of glaucoma disease. So up to now, we were able to disentangle that, yes, blood pressure variability relates to glaucoma, but we need, from a physiopathological perspective, a vascular biology of the disease, we need to disentangle this association of variability, and we observe that is the drops in the blood pressure due it to excessive blood pressure variability that relates to glaucoma. And the more likely hypothesis, because these drops in blood pressure, when they occur sporadically during the daytime or nighttime, they reduced ocular perfusion pressure. And I know that this was one-time 24-hour blood pressure monitoring, but imagine that your blood pressure for about 10 years, at least 2 times per day, is experiencing these drastic drops in blood pressure that is at least for seconds leading to ischemia or reducing the blood supply to the eye. That accumulation of drops in the blood pressure eventually should increase the risk of glaucoma, but that hypothesis we need to test in the future. But that's the reasoning behind when we look into the variability in the blood pressure. And now we have a bigger, and I believe, I hope so, a better picture of the vascular pathogenesis in glaucoma. And by the way, this variability, I know it's a simple, let's say, way to look at it. At least for glaucoma, it makes sense. And then you might think, okay, this is just for one disease. So what about, can we extend beyond one circulatory biological system? Because we were talking about the eye, but there are other circulatory systems. And this is what Professor Williger mentioned about that we are looking also into the brain. And I'm going to talk and describe about dementia and specifically Alzheimer's disease. So we know that Alzheimer's disease is described that there are certain pathological features of the disease. There is a atrophy of the brain. If you can compare here a normal brain with one with advanced Alzheimer's disease, there is reduction in the brain tissue, and specifically for Alzheimer's disease, there is hippocampus atrophy. There is accumulation of beta amyloid plates. These are the clinical features of Alzheimer's disease. But there is one hypothesis related to the development and progression of Alzheimer's disease that has to do with, as for the eye, with impaired brain blood flow and supply. So I told you for the eye that if there is an impairment to ensure blood supply to the eye, that will eventually lead to ischemia episodes of the optic nerve, leading to glaucoma disease anyway. Here is a similar hypothesis. And what happened is that the eye and the brain, they need to maintain constant blood flow. It cannot be interrupted. So in order to these circulatory systems to maintain their blood flow, they have autoregulatory mechanisms that in the case of that here, blood pressure goes too low or goes too high, the brain circulatory systems, as well as the eye, they're able to maintain a normal blood flow and supply. So that, like, okay, if the blood pressure goes down, doesn't matter, or if it goes up, if it goes up, we are still able to maintain a normal range. But not only this autoregulatory mechanism is impaired in glaucoma and Alzheimer's disease, but also if you add related to glaucoma, the hypothesis that high variability in blood pressure leading to drops in blood pressure causes impaired blood flow and supply to the brain that eventually should lead to Alzheimer's disease. And there is a lot of evidence, and they're really interesting nowadays that I noticed, to look into blood pressure variability in Alzheimer's disease. They have reported different, there are many papers actually reporting that the more variability during 24 hours and during the visit-to-visit blood pressure, the higher the risk of dementia and Alzheimer's disease. But that's all they reported. As it was done before, it's just based on the standard deviation, coefficient of variation, or mean. That direction of the association between variability and the brain, or in this case for Alzheimer's disease, has not been addressed. And they are thinking that, maybe the more variability relates to impairment in this blood supply to the brain, but they haven't been able to do, or they haven't done. I said they haven't been able because the evidence with 24-hour blood pressure monitoring is limited. So, of course, with that in mind, we cannot assess the 24-hour blood drops in blood pressure. So we're going to start looking, because we have this data in the Maracaibo agent study, and we started looking into blood pressure variability and brain outcomes, including microvascular damage and cognition. The first one is blood pressure and microvascular damage. But this project is in collaboration with a genetist from, that's Professor Williger, collaborates with that group, with a student, professor from Dr. Blanchero's department. And he's working on using, he's collaborating with us using the 24-hour blood pressure monitoring to apply his methods, which is based on the Warts distance. I'm not really familiar with this, but I believe it's a principal component analysis related method in which, based on statistical difference, generates different clusters. So what he's doing is to use the 24-hour blood pressure monitoring, and without having any other information on age or sex, he's just using that data. At this point, he was able to say that, in this population, there are 6 different kind of clusters. In a sense, there are 6 different kind of blood pressure behaviors or patterns, and this is the result that he gave us to us, cluster from 1 to 6. And this is using this Santa Rosa population, that is the one that has this, that they're a family, that has this genetic, I'll say, relationship, but still there's a lot of genetic variance there, but that's the population-based study. This is another 3D graphics in which he was able to say, we can still see that they are providing different information based on the method that he's applying. But eventually, after he gave us this, I was able to, based on the clinical relevance and the blood pressure levels, because that's what we use in the clinical practice, we were able to differentiate that out of those 6 clusters, there are 4 clusters that they are different. So, for example, cluster number 2, 6, and 5, we can group them in into only one group, and cluster 1, 4, and 3, they are separated ones. For example, if you only compare, you can see that cluster 1, their nighttime blood pressure increases more compared to cluster number 2. This is just some visual differences between the clusters, but with statistical significance, we said there are 4 distinct clusters based on the blood pressure variability. And then we wanted to see whether these clusters, they explain how blood pressure level relates to brain microvascular damage. This is a huge table with too much information. And what I wanted to tell you is that cluster number 2, 5, and 6, which we grouped them into one, this one's here, and cluster number 4, this one here, those participants with that cluster or that blood pressure pattern or behavior, those the blood pressure level at 24-hour daytime, nighttime, they were related to brain outcomes. This is brain artery stenosis, this is cerebral small vessel disease. What I wanted to tell you is that in this cluster and cluster number 4, high blood pressure levels, they were related to brain microvascular damage. We now Jose Garcia, that's his name, he's writing the manuscript. He's finishing writing the manuscript based on this data. And so far afterwards, we're gonna look into the variability in blood pressure, which these clusters are based on that variability. And by the way, we were able to replicate these clusters using the older cohort of the Maracaibo agent study, which was the Santa Lucia. And the second part of the blood pressure variability is that we are working with a mathematician from here at UTRGV, and she, in the Maracaibo data, the one that started in 1998, this is just an example. It doesn't mean that these were the exact dates we evaluated participants. But what I wanted to show is that there is longitudinal cognitive and 24-hour data. And what she was able to show is that during follow-up, if your blood pressure variability is like this, let's say this is just a follow-up, and then during follow-up it's just increasing and increasing more, she noticed and reported that an increase in the blood pressure variability over follow-up relates towards cognition or cognitive decline. And this kind of data has never been published, and that's what I referred before, that researchers have not been able to look into drops. It's just that this kind of data is limited nowadays, and this one, at least so far, I am aware that we are the only ones having this kind of data. So she reported this, and eventually her findings can be summarized or visualized as this. As we age, there's a natural course of cognitive decline. And if we have risk factors, hypertension, diabetes, environmental risk factors, they also relates to worse cognitive decline as we age. But what we are hypothesizing is showing that if you add blood pressure dysregulation, that is variability in the blood pressure over during follow-up, that increase or worsening of the blood pressure variability will also lead to a worse cognitive decline. And what we're going to do in the future, our hypothesis that this will also be related with a higher risk of dementia and related disorders. And you may guess, why is that relationship? I think I have mentioned before that similar to the eye is because this variability in the blood pressure and this project with Cristina from UTRGV, we were able to show that variability in standard deviation, or VIN, we use this time, relates to cognitive decline. But we looked, as we did for glaucoma, separately into drops in blood pressure. And we found that those drops in blood pressure and markers of low blood pressure, they were the ones related to a worse cognition. So eventually the hypothesis that as individuals age, these drastic drops in blood pressure, they occurred more, but they also carry a higher risk of cognitive decline. And the hypothesis behind is that during this, when this happened, these individuals, they're experiencing the brain artery regulations is not able to maintain the blood supply at this point, leading to, in this case, it's impaired dilatation or ischemia, but it's leading to damage in the brain microvasculature and brain tissue. And this chronically, over 10 years, might lead to an increase in Alzheimer's disease. We're proving that it's related to blood pressure dysregulation with cognitive decline, and this is what we're gonna do later on. So the variability in biological systems relates to diseases. Yes, there's a bunch of evidence about this. But so far, for the eye and for brain, for these outcomes, we have been able to show that it matters the direction of this variability. So if you remember before this first slide, I told you my experience, how the process of being involved in research has been, which was, there are certain points and I felt that, okay, I am a little bit lost, or am I doing the right thing, or Do I think that this is going to lead with something else? And so far, what I can tell in each of these steps is I initially started looking into 24-hour blood pressure monitoring. And then when I got my new topic about nocturnal blood pressure, because here I was looking into high blood pressure and then into hypertension, which is the main risk factor worldwide. And then I started looking at a new topic that was nocturnal low blood pressure. And I was like, well, but why if I always was looking into hypertension, now I'm looking into nocturnal low blood pressure? And that eventually led me to, there is something more. And it's not only blood pressure variability, nocturnal blood pressure drop, but also the variability. And now, why am I doing this? Because I know there is the variability in glaucoma and the brain. There is a direction of the variability. And what I would like to do in the future, what I'm trying to do also right now, is that to understand from a biological perspective, to be able to get some traits and phenotypes that will give me biological functioning of these eye and brain circulation systems to test a hypothesis, because now we know that there's an association brain, glaucoma, blood pressure, and glaucoma, blood pressure in the brain, but we need to give some traits and measure some functionality of the circulatory systems to fill also this hypothesis and continue doing research. And the same, during all this process, it has been important to explore. And this was key in this step, because I could have done my project, I could have been done with my project nocturnal blood pressure with policies, and that's it. And also, when I look into blood pressure variability, I remember I started my PhD and I wrote down because that was my first paper, variability, and I said, but there is something else. We should look into something that measures the drops in blood pressure. And I have to be honest, at the time, I didn't know how to do it. It took me 3 years after I started my PhD to be able to, and it might be simple statistics, or I really cannot tell, but in the beginning, I didn't know how to do it. But I knew from the start of my PhD that it was something that I needed to look into the future, and I kept that idea in my mind so far. And it has been able, of course, with collaborations with many mentors and also colleagues, and the experience of doing this also has been key to be able to achieve what we have achieved today, and I hope that we can working on this. So this is my acknowledgement that I have to thank Professor Gladys Maestre, who is the one who initiated the Melgarejo study, and since 2009 she has been my mentor, and also copy Professor Williger in your lead. And that's my time in Colombia. I really enjoyed that time and learned a lot, and since then and before as well, we have been collaborating together, and my mentor also from my PhD back in Belgium. So thank you guys for your attention. If you have any questions, I'm happy to answer.
Well, thank you, Jesus. And if you ask me, when we started this project, 35 years ago, 25 years ago, we were just collecting data. We started as a study of Alzheimer's. Who knew we were going to study glaucoma, right? Nobody. But what happened is that we built a study in Maracaibo, and we just kept adding traits because somebody in the Institute had an interest. Like Dr. Maestre shared an office with the Cardiovascular Institute, and they had these monitors. They wanted to look at 24-hour blood pressure variability. So she's like, yeah, do it here. And so you collect all these things over time. And then because you have a unique data set, you can ask really interesting questions. And Jesus has been doing a very good job of that, coming up with new ideas and new stimulus. So, we found a lot of interesting things in our research. But again, when you set up these projects, the most important result is the production of people like Jesus, who are given an opportunity to work with data they wouldn't have otherwise had access to build a career and to go, out from Venezuela into Belgium, into Colombia, and now to be a faculty member in Texas. So this is the real success stories, when you have a bright young guy like Jesus coming up with his own ideas and really building on stuff that we collected, not necessarily knowing why. He was able to put all these pieces together. So this is the purpose. And you guys are in North Africa are in a similar, situation to where we were in Venezuela, where you're trying to set up studies and build something to help young people have a data set for which they can build a career. And I think you've got a great opportunity, and Jesus is exactly the reason you do this sort of thing. Yeah, Yosher, come on, please chime in. Everybody, come on in and chime in.
Thank you, Joe. Thank you very much. Thank you for the nice talk. It was really very, very interesting and inspiring. My question is about the last step and the biological mechanism. Do you have a plan on how you will proceed to really work on this biological system and glaucoma in terms of how to really make use of all this data that you succeeded to collect? And as we are here in a genetic world, so is there any place to genetics in the plan that you are putting in place to really work on the biological mechanism?
All right. Thank you for your questions. So those are really interesting questions. And the first one is part of what I'm doing right now, what I came here. And what we're planning to do is for the eye, we are aiming to measure different traits of ocular blood flow and ocular microvasculature. So we're going to look into the retina microvasculature and measure structural traits, for example, the retinal vessels diameters or density. And we're also going to try to mention from a functional aspect. That one includes blood flow and for example, oxygen saturation as well. And we're trying to, in addition to right grants, also to get some, we already got some devices to measure OCT angiography that will give you the density of the retinal microcirculation and also some functional traits. And we are also going to we're trying to get some devices, for example, the Oxymat, in which I can see the saturation of the retinal microcirculation. So with those traits of functionality, the first hypothesis would be to show that there is an association between blood pressure variability and drops with impaired functional traits of retinal microcirculation, and also to test that the association between the mediation effect. Probably I should not use that word because from a statistical perspective means mediation means something compared something different, but we want to explain. We want to see whether the impairment in the ocular microcirculation due to blood pressure variability explains the progression, the development and progression of glaucoma. For the brain, because we're gonna also do it, we're gonna include trace of cerebral perfusion pressure. We're gonna try to do arterial spinning leveling, for example, and we're gonna try to measure with also functional aspect with PET scan as well. And eventually we want to test the similar hypothesis as we did for the for glaucoma. The only difference for this time, besides we're going to have these vascular traits and functionality of the brain, we're going to include cerebral circulation that will give us a structured picture of how blood pressure impacts the brain, because how the major arteries in the brain, the constitute of these arteries, the disease, the type of disease, mediates, and that we showed, we published, how blood pressure impacts the brain. So that's why we needed to include that into the equation. The second question, if I remember correctly, was about collection of the data, right?
It was more on genetics. So is there
a genetic one?
Yeah, maybe genes that are involved or associated with hypotension or I mean blood tension pressure. Sorry, if this can be studied to be like part of this I mean disease of glaucoma or any other disease that can be
For glaucoma, we're going to include genetics into this. We're planning to do, for example, the blood pressure variability and functional aspects of the eye. That's the next step that we would like to do in Maracaibo. And with that, we're going to collect samples to look into genetics. The limitation, which I don't know how to deal with, with glaucoma and genetics is that where we're gonna do the phenotyping, because the tissue, we can use bloods and then they will start arguing about the eye. But as I know, there's a genetic component in which you might have one eye experiencing drops in blood pressure that will not develop glaucoma. And then you will have another one who will develop glaucoma. So there should be a genetic susceptibility, yes, to the blood pressure impacting the eye microcirculation, leading to glaucoma risk. We're going to do the genetic phenotyping, and I know that there are some studies that they basically they when they do cataracts, they can take liquids from the eye. They can do some. They also do omics there. I have read some few studies, but that's for the eyes kind of the limitations. But we're going to do so because there's a genetic susceptibility there. That we are planning to do that as well.
Okay, thank you very much. Thank you.
Yeah.
So you mentioned several times that you have ongoing measures that you're trying to measure now, right, in these subjects. How easy is it to do these things today in Venezuela, given that we obviously have some difficulties?
Yeah, well,
I mean, it's a relevant topic. That's why I thought it was interesting to comment how it is, man.
Yeah. I've been actually not surprised, but when I left the country in 2019, it was really chaotic. And 2 years later, after I think the first COVID after the summer of the COVID outbreak, we evaluated more than 100 participants to do the eye examination. So, I was really happy that that they were able to do so, and I think one of the main parts as people is compromise. They're willing to collaborate with us in order to, for that time, it was the Eye Institute, and they say, we'll try to make it. With all our efforts, we're trying to make it. We're going to bring participants here. We're gonna do these protocols, and we're going to be able, following all these, because of the COVID situation, the Venezuelan situation as well, we're going to do these evaluations. And also there was the community involvement as well. Because sometimes, why they will go to another part, and that's, I don't know, at the time, the whole perception about COVID, but there was the situation, but still, I know that for Santa Lucia, Santa Rosa population, there is a great involvement from their community, for the community, in order to participate in the project. And I believe part of that is that the way that since the beginning, we have been able to be transparent, and we have been able to communicate when we are able to with the community. And there's also in Santa Rosa, there is a community center, and that has been an extremely important link between us and the community. So they are aware of what we're doing, what we're gonna do, so they feel they feel safe. That's okay. If you're gonna come and do evaluations here, I'm up to it. So please let me know what we should do. That's the community part, which I don't know if that happens everywhere. Probably in the United States, there have been a little bit more resistance, but there, in summary, I would say there's the team involvement and also the community involvement so far that they have given us a chance to conduct this studies. And that was in 2020 or 21 that it was possible, and we did it. So that was great.
Yeah, I think that's like one of the most important things is really you have a successful study because of all the effort that was put into building that relationship with the community and having it so that the patients and the people involved feel like they're part of it. It's like we told them they may not get any real benefit out of participating, but we explained it was for science and all this, and they build trust with us. And that's the key in any country. And it's the same for when you guys set up things as your model when you go forward and do this stuff in North Africa. Yeah. Joe, do you have some comments or questions?
My comment is that going back to the question that Yosser raised, I think I think Jesus has done a fantastic job of setting up a pre-stage of genetic study in that, number one, he's further developing families in isolated inbred families. And also he has a general population of randomly selected sample from a community so that he can use pedigrees, isolated pedigrees to identify possible candidate genes to characterize. And then once you identify and characterize those genes, you can look at the general population to see what is the impact in the general population. So I think his setup is fantastic, and he has done tremendous work. And he and Dr. Maestri, the PI, has done tremendous, and Terwilliger is also co-PI, has done fantastic job of biobanking these things so that when the technology is right, then you'd be able to use it. Now, second, even more important thing is that what Jesus has done is this. When you think about glaucoma and hypertension, we all know that it's an incredibly multifactorial disease and it's genetically heterogeneous. So that if you just simply look at the relationship between the genotype and phenotype with the hypertension or glaucoma as an endpoint, you're not going to find anything interesting. It will be very difficult to find anything interesting. However, what Jesus has done is that he has characterized a unique properties where the 24 hours of blood pressure is dipping, and these are the special trait that you want to go after so that it be more specific, so that when you're looking at genotype-phenotype relationship in a multifactorial disease, he may be able to find a gene that contribute to that particular phenotype, subphenotype, that may eventually lead to hypertension and other things that mess up your brain. So I think he has done tremendous advances in the understanding of how your biological system works in Venezuelan population. But you can apply the same principle to see whether does the same thing happen in other US white population or in Tunisian population, and that's something to look at. So I think these first, when we think about genotyping, most people just think about sequencing the heck out of everything biosample you have, which I think is not a best way to invest your limited resource. I think first characterizing phenotype that appears to be genetically or biologically oriented is the first thing so that you understand the phenotype better. And then once you do that, then genetic study becomes far more efficient and easy. So I think the first step he's working on is the right direction and going in and leading to increasing the likelihood of success in the research. So that's what I wanted to add.
I would also add that Jesus mentioned this, but didn't really focus on it, is that one of the two populations in Santa Rosa is one family of 5,000 people or 8,000 people or however many thousand people. And so right there, you have a unique opportunity to look at the genetics of things like 24-hour blood pressure variability, right? So that's a whole other level of things that can be done in the future. Now, he's first trying to demonstrate that this is something really important for disease. So now that we've established the importance of this trait, now we can go look at the biology of the trait. What causes that, right? Why do these people have this pattern? Like when you look at all the different sort of trajectories of blood pressure, why is this one there? And what is the reason? He's looking at the reason that this affects disease, but then the genetic question is, well, why is it like that, right? That's a different question, right? And that's, I think, something that clearly can be investigated in the future since we have the samples from most of the people in this population and access to it. So it's pretty easy to do that later, but that's a totally different type of research than what Jesus has been doing, which is to find these quantitative risk factors, biological traits that are actually perhaps more important to study than the disease itself, and maybe more simple in their etiology. Yeah? So, I mean, what do you think, Jesus, about all this stuff?
Yeah, Yes, there is. Wait a minute. How can I? In a slide. Oh, that's why. So, yes, you are right. Let me, I'm doing something here. There you go. That when we in the last slide, I referred to blood pressure dysregulations. And the hypothesis that over 24 hours, those who are experiencing a lot of variability in the blood pressure that is leading to drops in blood pressure, etc., that relates to eye and brain damage. But something interesting here that so far they call it a limitation of the ambulatory blood pressure monitoring, which I believe is true. It's a limitation. But they call is that if I want to do a longitudinal study, I would say, for example, I would say that variability changes a lot here, and then it doesn't change during the rest of the day. This is just an example A. And then what they are hypothesizing is that if I do a second ABPM, a 24-hour blood pressure monitoring, it should be replicated the pattern that I found in my previous ABPM. So, for example, the nocturnal deepening blood pressure, they don't believe it. They don't believe in this so much because they are not able to reproduce in a second ABPM the variability. So what they have found is that the variability where it was supposed to changes a lot, it didn't occur. It will occur eventually in a different period of the day, but it didn't occur in the beginning. And because it didn't occur in the beginning, then they say the method is not feasible. I understand that patients' daily activity, participants' daily activity can change every day. Sometimes I might go to a supermarket and then the other day I didn't go, so that's why it changes during the daytime. So it can actually do that. But if we're looking into the extreme part of the blood pressure variability, as Professor Joe specified, that's traits that indicates that there's an abnormal pattern of the blood pressure that relates to disease. What we're going to be able is to, when we apply the genotypes or genetic analysis here, is to see what are the genes that are causing these drastic drops in blood pressure that they relate to the disease. And eventually, my hypothesis is that whatever we found here, it actually is going to be different in the next evaluation. I would not expect that the blood pressure variability will occur at the exact same time, because whatever that gene is related with impaired or a normal circadian rhythm, it would just mean that it can occur at any time of the day. So these kind of questions about, and that's why it was important having a longitudinal ABPM, it has to do with the abnormal circadian rhythm, and that's what we are referring to blood pressure dysregulations. So what I believe that's once, as described Professor Jolie, and also you too, you're totally here, is that once we identify these traits, baseline and longitudinally, whatever specific gene that is, that gene, every time we evaluate blood pressure, it will be totally different. It will be a mess. I would not expect that there is a pattern, and therefore in the clinical practice, we will say, it doesn't matter. It was not replicable. So it's not as good as it should be. But this gene represents a normal circadian rhythm. This is what is going to happen. That's why I believe it's going to happen. We're going to have these traits indicating abnormal patterns in the blood pressure as well. And that's why I think also it was really important, the work that this person from the Dr. Melgarejo group is doing, is that he really doesn't care about the level, which is what we use in clinical practice, is that the patterns, the clusters of blood pressure variability. So I believe that also there could be something that we can link with blood pressure. And there is something I didn't mention to you because of time, but you see 6 patterns. These patterns were obtained in the Santa Rosa population. That is the family study, right? He was interested in this because he has the irritability of this population. So there is that trait. he identified these 6 patterns, and then he told me the only one that we were able to replicate to show that there is an irritability trait was cluster number 1. So out of the 5 clusters, out of the 6 clusters, the only ones that is irritable is cluster number 1. And we thought, oh, okay, it might not matter much. But then what I noticed is that when I started looking into the baseline characteristics of these patients, these participants, cluster number 1, although the base age, sex, body mass index, everything is the same in these 6 clusters, cluster number 1 has the lowest blood pressure levels. In a sense, although it's the same age average compared to the other ones, this group of individuals, they have the lowest blood pressure. And not only that, when we look into clusters separately, which I didn't show, we found that temporal and occipital white matter hyperintensities, that's a kind of serious small vessel disease, this one here. The white matter hyperintensities, cluster number 1 has the lowest burden of cerebral small vessel disease compared to the other clusters. So far, we are not including that in the manuscript because we feel that we need to give a little bit more of thinking about this. But in conclusion, like this cluster number 1, the pattern of the blood pressure was irritable. And not only that, these participants, they have the lowest blood pressure and they have the lowest burden of cerebral small vessel disease. So in conclusion, this irritable trait, we can call it a protective trait. It has a different pattern in the blood pressure compared to the other ones. But this group of individuals, they have the lowest blood pressure and the lowest burden of certain markers of cerebral small disease. So that's I think I should have mentioned that before, as just to give you some information about the genetics part about this. But based on this, that there's an irritability pattern in the blood pressure. If you look into circadian rhythms, I think one of these traits or the identification of these clusters traits will give us a chance to relate them with genotype genetics. And of course, have a better understanding, at least. And I'm still curious why this cluster, same age, same everything, they have the lowest blood pressure. I'm still really curious about this. But yes, that's something I wanted to add.
One other question I had is like, in order to measure this, these people have to like wear some device that physically does something every so often whenever they measure the blood pressure, right?
Yes.
So that presumably affects their daily activity. And it also affects sleep. It affects, if you have this thing on your arm, you may not want to do certain things. So how is that like how do you deal with that potential covariate?
Yes. I hope that, well, this one briefly in the future, but they are doing it now, but I don't believe they're still validated. People don't believe they're still validated. They are making cuffless devices to measure blood pressure. We don't know how they do it because it's all patent, but probably it's, I don't know, signaling of the radial pulse, for example, it could be. But what they're trying to do is a smartwatch, for example, or something that is more convenient to work during 24 hours or days, because they are also interested in measuring every week, etc. So I believe in the future, once we are able to, yes, this is within the experimental world, this is something that can change, that can influence the blood pressure patterns. So we hope that there's going to be a cuffless device that is able to measure blood pressure, not to estimate blood pressure based on many parameters. That's what we hope. And for us, although yes, indeed, the devices they affect. If I take a blood pressure now, as I mentioned before, I went to the supermarket, but then next day I didn't go in the morning, but I went in the afternoon, for example. That we changed the pattern of the blood pressure. But so far, when we put data, all these of all these participants, we have a diary, but let's forget about that diary. When we pull this information, we have been able to show that there are physiological patterns. There is a normal variability in the blood pressure during the daytime. There is a nocturnal dipping in the blood pressure, as we can see here. And there is a morning surge in blood pressure. And this is when pooled data of over 10,000 participants from the general population has been included into one database, which is the research work we were doing before, we still doing with Professor Jan Stassen, if you remember him. And so in all these 13,000 participants, they are from the general population, so eventually they are not patients. So they are participants, so they do their normal daily activities. And so far, even with that, we have been able to identify that if they do not follow these patterns, that is, if the nocturnal blood pressure actually increases or there is a too much variability, that relates to cardiovascular outcomes. So we need to treat the other one of their daily activities, what is going to happen. But then that part, we show that there is a pattern. And if we go beyond that into drops in blood pressure or the clusters that Jose Garcia is doing, if we're able to relate that with disease, in this case, that's at least we should have a clue where to start or which parameters to take into account. I also believe that if you are, even if you're going to the supermarket or if you have a fight with someone, if your blood pressure increases too much, that if you accumulate that peaks in blood pressure, for example, that should not be good for your heart. And if you are able to show that whatever reason it was, those peaks relate to cardiovascular outcomes, then we should look at them differently. And the same for drops in blood pressure. If there is something that is causing, which likely is not because of daily activities, but whatever is causing drops in blood pressure and that relates to disease, we should be able then to identify patients' behavior to understand why those happen. And that's why we have the diary, but I still don't know how we can manage that because the diary is not really specific just for treatment, but just the conclusion, that might be a limitation, but we are able to replicate that patterns in a large cohort. And I hope that in the future, the cuffless devices stay with this, we were able to tackle some of these issues.
Well, before we wrap this up, I mean, we're going to have a bunch of people are going to be viewing this, offline because it's a holiday now in North Africa. But you're a young scientist. You've done your medical school in Venezuela, and you've done your PhD now in Europe, and you're now got your assistant professorship. Do you have any advice for these other young scientists who are starting their careers now and they're working in North Africa and so forth about what did you learn? What do you think is your best suggestions for people that are getting started? I mean, you've just gone through it. So, yeah.
Yes. I do know that for everyone's circumstances is different, conditions are different as well. I believe in my case, my experience, despite all the bad circumstances of being in Venezuela, being part of a low-income family, I think that's regardless of the necessity, what was important to me is that I kept insisting and I waited and kept working on what I wanted to do. I didn't have and still don't have a clear idea of what is the main hypothesis I will discover, what will be my major contributions. But what I did know is that this is something that I liked, they still like doing. I needed to feel passionate about what I was doing because the only thing that drove me apart to, just working in order to provide, for example, for your family is that, okay, everyone's circumstances is different, but kept motivated, passionate about it, and time. It really took some time after I started seeing that I am making some progress from a scientific perspective. Also, that's I whatever motivation I was showing, that was something that my supervisors that also include you guys, saw me and gave me the opportunity to work in certain projects. So I believe we need the academic part, we need the education, we need the skills, that's important. But whatever place I went to, I was always motivated to do what I was asked to do and beyond that as well. So it doesn't matter the hours that it took me, I was always really passionate and motivated. I'm going to do this and I'm going to do more. But I'm not going to do more and just workaholic and everything. It's just that really motivated. That's, oh, I did this, but what if I do this? And keep thinking. And that's the exploring part. That even when I went back home, I was still constantly thinking, okay, maybe I should do this. And because I was interested in it. So patient, time, it really took some time to get me where I am today, which I didn't see 10 years ago. And probably 10 years ago, I should have decided to do something else because of my circumstances. That would have been the best choice for me. But okay, here I am today, and I think, okay, it really requires a lot of patience and just motivation that good things will come and still are going to keep coming. And I have I don't know why, I have a genuine interest in science. So that motivation makes that everything is not only about work, it's just making science. And that part also was important to me. And mentorship, I think having a good mentor matters the most of what that institution is. And that mentor will give you opportunities and will teach you, but also will put you in the right place in which you can start walking towards whatever goal it is. So that also is important to find a good mentor as well, and collaborators that you know that you can count on. And if you don't know, well, they're gonna take you and they're gonna try to at least show you what you should do in order to learn. And I think so far that's what I can tell. Yeah, and it has been a long process, and it's gonna be a long process too. I mean, there's still a lot.
Well, I guess we're supposed to wrap up. It's around 11:30, but I really want to thank you for taking time to come give this talk. I think you've done a lot of really interesting work, and I'm hoping that we'll continue to be able to work together in the future and develop some more ideas. And anyway, thank you all. And we hope to see you all next month when we give the 19th lecture in this series. So thank you all.
Thank you very much. Really happening. Bye-bye. Have a nice day.
Thank you, Joe. Thank you.
Thank you.
Thank you all. Bye-bye.
Bye.